Hi everyone, my name is Annika, and today I'll be talking about pericyte coverage on microvasculature in the aging spinal cord. So what are pericytes and why are they important? Pericytes are a type of cell that reside on microvessels, and they play a very important role in regulating blood flow, as well as maintaining the blood-brain barrier and the blood-spinal cord barrier. Currently, a lot of research is focused on the brain, and even though they're both part of the central nervous system, we can't assume that pericyte coverage is the same in the spinal cord as it is in the brain. There's also very limited research on how, if at all, pericyte coverage changes across one's lifespan, especially in the spinal cord. If you look at this side view on the left, you can see a capillary, and it has several pericytes denoted in blue. There are other microvessels called arterioles branching off of the capillary. You may notice that the arterioles have smooth muscle wrapped around them, whereas the capillary does not. This means that the role pericytes play in vasoconstriction and vasodilation of these microvessels is even more important, because the capillaries don't have that smooth muscle to fall back on. If you look on the right here, we can see a cross-sectional view of the same capillary with the pericyte, again in blue, wrapped around the vessel. You may also notice that the pericyte is working in conjunction with astrocytes or astroglia and microglia as well. But it's important to note that the pericyte is the only type of cell that makes direct contact with the capillary. And I'll talk more about astroglia and microglia later. We know a little bit about aging or a little bit about how pericytes and the relationship with microvasculature changes as we age. The first thing we know is that there's a decreased microvessel density and increased tortuosity as people age. And tortuosity just refers to essentially how bendy the blood vessels are. If you look at this image here on the right, we can see that in the young brain, there's a lot more bidirectional blood flow, shown by the red and the orange in this ultrasound image. And we can also see that the blood vessels are very well defined, whereas on the right, we see an aged brain where blood flow is primarily in one direction, and the blood vessels are thinner and not as well defined. With aging, we also see a loss in pericyte coverage of the blood-brain barrier, which can lead to a deterioration of capillary flow as well as limited oxygenation of brain tissue, which can lead to a host of problems. With aging, we also see increased blood-brain barrier and blood-spinal cord barrier permeability, which may be in part because of a loss in pericyte coverage. Based on what we know so far, we hypothesize that for this project, pericyte coverage would be lower in our age group than our younger group for both white and gray matter. The aim of this project was to analyze pericyte coverage in female rat cervical spinal cord tissue. We had a cohort of 6 female rats, 3 young and 3 aged. Our young rats were 3 to 5 months old, whereas our aged rats were 19 to 21 months old. We began the project by sectioning cervical spinal cord tissue into 20-micron slices, and then used immunohistochemistry, which a few of my peers have talked about so far, to stain for different cell types. We used lectin to visualize blood vessels, and G2 for pericytes, GFAP for astroglia, and IBA1 for microglia. We then imaged these tissue samples using an axiozoom microscope. And after taking these images, we imported them into ImageJ and quantified the pericytes manually, which essentially meant zooming in, adjusting brightness, contrast of the image, and manually measuring each blood vessel and counting the pericytes on them. And for this project, we had about 3,000 vessel measurements. Finally, we analyzed this data using Google Sheets, Microsoft Excel, and GraphPad, and calculated pericyte coverage as a percentage using the formula of the number of pericytes divided by the length of the blood vessel multiplied by 100. On this slide, we can see a comparison of gray matter tissue between our young and our aged group. On the far left in blue, we see the DAPI channel, which stands for nuclei of all cell types. And then we see NG2 in red, which again stands for the pericytes, lectin in green, which stands for the blood vessels. And on the far right, we can see a composite image, and where we see all 3 channels, the blue, the red, and the green, that's where we have pericytes. So there are more than 4 pericytes in each of these images, but I've just circled some of the most obvious ones here in the young and the aged group. Same thing on this slide, except it's a comparison between it's a comparison for white matter between the young and the age group. You may notice that there are several pericytes in these images as well, But for the white matter, we generally had fewer longer vessels, whereas the gray matter had more shorter vessels. And these are just more pericytes. Let's see. Our results were somewhat surprising in that we didn't see a very big difference between pericyte coverage for the young and the age group. Coverage was almost exactly the same without looking at the different types of matter. But we did see some differences when we separated between gray and white matter. Surprisingly, we saw that there was a statistically significantly higher pericyte coverage percentage for the age group for gray matter, whereas as we expected, the young group had a higher pericyte coverage for white matter. Oh, and these results are somewhat limited in that we only had 6 animals included in this study. This project is ongoing, so these results may change as more data is collected. While I mainly focused on pericytes this summer, I've been working very closely with Dr. Preecha Chandran, who has begun to look at astroglia and microglia as well. Doing a preliminary analysis, we found that our age group has had much higher astroglia activation than the young group. And this is a different scale. It's intensity instead of counting the number of cells. And you can kind of see those differences where the age group is a little brighter in this image than the young group. And then we saw the same thing for microglia, where there's more intensity, higher activation for the age group than the younger group. So in conclusion, pericyte coverage was lower in the age group for white matter only, but these results are limited by our small sample size, so we'll continue to collect data on pericytes in the future. Astroglia and microglia activation was also higher in the age group, but again, those results may change as we continue to collect more data. I would like to extend a big thank you to everyone in the Khaing lab, especially to Dr. Xing Khaing and several of my wonderful mentors, Dr. Preecha Chandran, Lindsay Cates, Jeffrey Hyde, and everyone on this slide has played such an instrumental role in making this a wonderful experience for me. And I would also like to thank the Ellenbogen family, Julia, Sylvia, all of our donors who have played really key roles in making this program happen. So thank you all very much.